Hello everyone. Welcome to another discussion from Whole Pharmacy. The today's topic of our discussion is uh, cellular mechanism or the deep mechanism of apoptosis. Basically, there are two cellular mechanism of apoptosis. The first mechanism is mitochondrial or intrinsic pathway. Mitochondrial mitochondrial which is also called as intrinsic pathway. Intrinsic pathway. Okay, this is the first mechanism. Definitely we will be discussing it later on. The second mechanism is death receptor pathway. Death receptor pathway. Death receptor pathway. Which is extrinsic in nature. Inshallah, we will discuss this death receptor pathway in next episode. Extrinsic. Now come towards the mitochondrial pathway, which is intrinsic in nature. Now the question here arises that why cell wants apoptosis of itself. So there are certain reasons. For example, cell injury. When there is any injury which is not going to be repaired by the cell repair mechanism, definitely cell wants itself undergo apoptosis. Next is DNA damage. When the DNA damage to the cell happens due to any reason which is not repairable by the proteins, for example P53 pathway, then definitely cell again wants its apoptosis because cell says I am not required anymore. The next is protein misfolding. When due to any reason there is misfolding of the protein, cell wants itself to be removed from the system. The next reason is loss of growth factors. Because basically growth factors are required for the survival of the cell. When due to any reason these growth factors becomes deficient in the cell says itself that I am not required anymore, I should be removed from the system. So in this way apoptosis of the cell starts. Okay, now come towards the mitochondrial intrinsic pathway. First, when due to these reasons any reasons happens, there is the activation of certain gene in the cell which is going to be apoptosed. The name of those genes are BIM gene, the second gene which is BID, the third gene is BAD. These three genes are involved in the process of the apoptosis. These are basically genes. What happens next? These are genes, these then translate proteins. Those are also called as BIM protein, BID protein and BAD protein. So these proteins then formed. After that, these BIM, BID and BAD proteins causes the activation of some other proteins, and the name of those proteins are Bax protein and BAC protein. These proteins are basically a pro-apoptotic in nature. Pro-apoptotic apoptotic in nature. So what these bags and bag genes or proteins basically going to do? They basically inserted into the mitochondria. These proteins goes into mitochondria of the cell. Mitochondria of cell and increase the permeability of mitochondria. So when the permeability of the mitochondria increases, means the mitochondria becomes fluffy and leaky in nature. So that's why it starts releasing certain enzymes. It becomes leachy, it becomes porous in nature, and it starts releasing different chemicals, and one of those chemicals is cytochrome C, which is cytochrome C. So this cytochrome C, which is released from the mitochondria because of its increased permeability, this cytochrome C then binds with a protein that is called as APAF, apoptotic factor 1. This is a factor which is being activated or triggered by this cytochrome C, which is released from the mitochondria. Now then this apoptotic factor 1 activates the group of enzyme, and from one of those group of enzymes is Cas-Pas9. It becomes activated because of apoptotic factor 1 activated. Okay, after that, after the activation of Cas-Pas9, this Cas-Pas9 again activates its enzyme which is similar to Cas-Pas9, and the name of enzyme is Cas-Pas3, which is the enzyme which is similar to the group of the Cas-Pases. Cas-Pas3. This Cas-Pas3 becomes activated by the Cas-Pas9 activated. Okay, after that, when the Cas-Pas3 becomes active, it performs two functions. First, it inactivate DNA repairing enzyme. So this is the first function that is performed by this Cas-Pas3. This is the first function. The second function is that it activates DNases DNases. Those enzymes, those causes the fragmentation of DNA. Fragmentation of DNA, because cell knows that it is not going to be divided anymore, so that's why caspasis causes the breakdown or chopping of DNA or the fragmentation of the DNA, because this cell is not required anymore. So when these two processes happens, inactivation of the DNA repairing enzyme and activation of the DNases, the cell on its surface starts producing certain signals, and those signals are called as eat me up signal, because the cell says that I am not useful anymore, please eat me and remove me from the system. So when the eat me up signals is generated from the cell, the phagocytotic pathway of the body become active, which is performed by phagocytes, and these phagocytes are monocytes and macrophages and the dendritic cells, etc. So in this way, there is the chopping of the cell happens. The cell is converted into fragments, and it is being eaten by these phagocytes like monocytes, macrophages, and related cells like dendritic cells and other cells. So when these monocytes, macrophages become active, they causes the phagocytosis and causes the fragmentation of the cell and send all the chopped material to the lymphatic system, and this lymphatic system then drains into the GIT and removes the whole that apoptotic cell from the body. So this is the very controlled pathway of apoptosis, in which there is no inflammation to the neighboring cells. Means if we have these three cells, cell number 1, cell number 2 and cell number 3, and cell number 2 is going to be apoptosed, cell number 1 and 3 even will not know what is happening around cell 2. So this is very controlled and tight process of apoptosis, and it is associated with no inflammation. No inflammation is associated with apoptosis pathway. So let's recap mechanism of apoptosis. There are two mechanisms of apoptosis. The first one is mitochondrial or the intrinsic pathway. The second one is death receptor pathway, which is extrinsic in nature. We will definitely be discussing in the upcoming lectures. Okay, come towards the mitochondrial and intrinsic pathway. When cell injury, cell damage, protein misfolding, loss of growth factor happens, there is the activation of BIM, BIT and BAT genes. These then translate the BIM, BIT and BAT proteins. These proteins then activates. Sorry, I forgot to write. Activate these back protein, which are basically pro-apoptotic protein in nature. This goes into the mitochondria of the cell and increase the permeability of the mitochondria. When the permeability of the this is the process, or the process number 1. In the process number 2, these pro-apoptotic backs and back protein goes into the mitochondria and releases. This is process number 3. Then releases the cytochrome C. This cytochrome C then binds with the apoptotic factor 1. This is process number 4. Then it activates the Cas9, which is process number 5, and then it activates the Cas3, which is process number 6. and it Caspase causes the inactivation of the DNA repairing enzyme, because this cell is not going to be survive anymore. So the second function is activation of the DNAs, which causes the fragmentation of the DNA. So eat me up signal is generated from the cell, and when the eat me up signal is generated, the phagocytotic pathway of the body becomes activated, and in the phagocytotic pathway, monocytes, macrophages, dendritic cells, natural killer cells, and all those cells involved in the process of the phagocytosis, because the phagocytosis of that apoptosis cell. So in this way, apoptosis of a particular cell happen due to these four reasons. Reason number 1, reason number 2 that I have already explained in the start of the lecture, reason number 3, and reason number 4. I hope you enjoyed this lecture. Please, if you like it, please spread it to your neighboring, and like, subscribe, and share. Thank you very much.